Welcome to the OCCPR webinar series. Today we will be discussing a heat map visualization tool from the CPTAC program called Protrack. This webinar is hosted by Biomedical Software Engineer Anna Calinawan of the Ichan School of Medicine at Mount Sinai, New York, and Dr. Pei Wang's lab. All registrants are muted but can use the chat box to send questions to the speaker, which we will try to address during the question and answer period at the end of the presentation. This webinar will be recorded and made available on our website. Thank you for joining.
Thank you for joining the NCI's Office of Cancer Clinical Proteomic Research webinar series. As a part of NCI's Cancer Continuum, OCCPR mission is to improve prevention, early detection, diagnosis, and treatment of cancer by enhancing the understanding of the underlying molecular mechanisms while advancing proteome and proteogenome science and technology development. Through the integration of genomics and transcriptomics with proteomics, also known as comprehensive proteogenomics, OCCPR's aim is to advance basic, translational, and clinical research through OCCPR-supported programs. This webinar series hopes to educate you to all the resources OCCPR and its researchers have to offer.
As Dawn mentioned, I'm Anna Calinawan. I am a software engineer working at Mount Sinai with Pei Wang and Boris Riva. Today I'm presenting a tool that was built under the auspices of the CPTAC consortium called Protrack. I'll be presenting the first prototype iteration of it, which was developed for clear cell renal cell carcinoma. So this is why it's Protrack CCRCC. And this is essentially a web tool that's an interactive multi-omics data browser. So it'll allow you to see different types of data for specific proteogenomic cancer studies. And so I'm going to go ahead and talk a little bit about Protrack and why we developed this tool. The CPTAC consortium wants to understand cancer across all of its molecular profiles, and that includes across the entire cancer continuum, and that includes across the central dogma of biology, where DNA, which has modifications, is transcribed to RNA, that's translated to protein, and that produces metabolites. And so to this end, along every part of this spectrum, the CPTAC has produced associated data for specific cancer types. So we have for whole exome, whole genome DNA methylome data, transcriptome data, proteome and phosphoproteome data, and for some cancer types, even metabolomics data. But then, of course, the resulting question is, how do we see and understand this multi-omic data together? How do you digest such large data sets in ways that can readily show you patterns without necessarily having to know how to code or looking so hard at an Excel sheet that your eyes start to water? And so for one of the first studies that we performed as part of CPTAC 3, one of the first papers to come out was the clear cell renal cell carcinoma study. And this is already available and published in Cell. And the way that they show all of this multi-omic data together, one of the ways is through a really nice heat map. And this is great because you can see all of these different types of data together in a way that's a lot more digestible. You have mutation data, methylation, RNA, protein, phosphocyte data, phosphocyte abundance, as well as copy number and chromosome level data and some clinical data and clinical annotations. And so this is like a great succinct way of condensing a lot of points of information together. But there are, of course, some limitations. And so that's why we decided to create an interactive version of this kind of data visualization. And so I want to take a little closer look at the specific figure from that paper to talk about what the limitations are. So if we look at this figure, the caption reads, it's a heat map of multi-omic data for the 5 key tumor suppressor genes associated with kidney cancer, with clear cell renal cell carcinoma, VHL, PBRM1, BAP1, SETD2, and KDM5C across 103 samples, and the tumor samples were ordered by 3p CNV alteration from loss to neutral. And so I want to talk about some of the limitations by going through this caption. Specifically, if you have 103 samples, let's say, for example, that we are interested in what is underlying one sample. If we see this particular sample that doesn't have any 3p CNV alteration, and it's the only CCRCC case that doesn't have that, how do we find out what is actually underlying that sample, which sample it is, maybe some clinical data about it or histological images? That's impossible with a static heat map. Here they chose very clear marker genes for this tumor type. So the issue is you become limited to the set of genes that the authors have decided are worth focusing on. And finally, the tumor samples are ordered in this case by the 3p CNV alteration, because this is a very vital thing in clear cell renal cell carcinoma. But you might want to be looking for a specific or different set of patterns. For example, but if you wanted to look at how the expression of HRAS corresponded to the increase in stage or grade. And so the question is, how do you customize a visualization like this to serve your own needs? There are only really two ways that I can think of it, and they both entail downloading and accessing the data. And the first way would be, once you have the data set, using it to code and create your own heat map. And the next option would be to upload it to a more general platform, which can ostensibly generate heat maps for you. There are limitations to both these approaches. For the coding one, you'd have to know how to code. And so that already limits who has access. In the second case, there's always a fear when transferring data from one store to another of some sort of untraceable transition between the two of them as it moves along. And there is also just the issue of wasting time and space with respect to time to upload, download data, and the redundant storage space. And so this is why we decided to create Protrack, which is a web tool that means you can access the data from any modern browser and just look around without having to pass this data up to another visualization platform. You can just interact with it right there. And so I'm going to show a live demo of some of the features of this tool. This is the user interface for the tool for CCRCC. The tumor type is here prefacing CPTACdataView.org, which is the URL. It's very, very simple and intentionally so. This is kind of like a starter kit to the more complex tools that I think can be quite intimidating. This is very straightforward: input, output, interact, where you have here a section where you input the genes of interest. Right now, I've put in the genes that were actually included in the CCRCC paper. You simply click Visualize. You can download the image for use later, and you can also download the Excel spreadsheet. And so I wanted to show some of the interactive features to start here, which is we were talking about this one sample, CCRCC sample, that had no 3p CNV alteration. And so when you click that, you can see the underlying data, including the individual sample ID. We actually link out to the histologic images organized by tissue type. They go to the Cancer Imaging Archive images. And so maybe a pathologist would like to know, could see right away there's something that seems to be different about this specific sample. You can see some of the clinical and demographic data that's being pulled down from the Proteomics data commons. That's an ever-growing API of annotations. And so I'm pulling directly from that, and this is live data. So if there's anything that anybody decides later we want to add on, please feel free to let me know if it feels like it would be beneficial to know this kind of data about the individual samples. And so that's just an example of the kinds of things that you can see. It becomes sort of a one-stop shop while you are looking on this web browser for this particular data set. So I wanted to show another specific set of data here so we can customize the genes that we want to see. This list of genes is chosen because they are a series of immune markers, so CD8A, PDCD1, PD274, and so on. I'm highlighting this because one of the major findings or major areas of study in the kidney cancer study was the immune groupings associated with the cancer. And these immune groupings are CD8- inflamed, CD8+ inflamed, metabolic immune desert, and VEGF immune desert. And so when we visualize these genes, you simply enter the genes there and click Visualize, and a heat map will render. So I'm really interested in the immune system, and I would like to understand what is actually the molecular underpinnings of this immune grouping, or what is actually going on at some of the molecular level for some of these immune groupings. And so all I did here is you select a track, and then you can sort the track by ascending or descending. And then the rest of the track will sort according to that. And that way you can explore and develop the patterns that you are most interested in. And so in this case, I sorted by immune group so that I can see all of the different molecular markers grouped together by these specific immune groups that I'm interested in. And based on this, I can see from the immune grouping that CD8A is quite enriched, the RNA expression is quite enriched in the CD8 plus inflamed immune group. And that confirms that this immune grouping has some validity to me. I can see that it's the same case here with PDCD1. I'm not entirely sure what PDCD1 does. I want to know some more details about it. You can actually click the G name when you enter it. A summary from the NCBI is pulled down, and you can link out to more information about the NCBI. So in this way, this tool also becomes sort of a one-stop shop for, let's say if you wanted to find a particular molecular pathway and to copy those genes into the gene input, and then you wanted to understand, I see a pattern here, what's the fastest way for me to understand what this particular gene is doing in this pathway? So I'm trying to look at RNA data, and I'm not too interested in copy number variation because it's a little distracting. I'm going to actually deselect that, and I want to show that feature where you can toggle tracks on and off. It allows you to tailor the experience in a way that allows you to focus on the information that you would like to see. Okay, and so I also wanted to see here, if I see VEGF, it looks like, oh yes, the VEGF-A RNA expression is also enriched in the VEGF immune desert. So let's sort that RNA expression by desequencing. Okay, great. It's been sorted in descending according to these secondary groups that have been sorted. So we can really see here the number of samples that are highly enriched in VEGF-A RNA expression in the VEGF immune desert immune group. And so this is ultimately an example of the very straightforward but hopefully very practical things you can do with this particular tool. Okay, so we've ordered this in a way that we find meaningful. If you download the Excel form, the Excel sheet, I'll show you what you actually get when you are trying to pull this data out. So specifically, you get those samples ordered in exactly the way that you determined was an interesting pattern for you. And then the same clinical tracks that you have here, the individual genes marked as separated by their data type and the corresponding values. You can find it at ccrcc.cptacdataview.org. It is entirely open source, and so you can see the source code on GitHub. It has been submitted to a special issue of Proteomics entitled Data and Systems Biology and has also been deposited in bioRxiv. This is, as I mentioned, part of the whole CPTAC effort, and so I wanted to thank the members of the CPTAC Consortium, the OCCPR, the people at the NCI, my colleagues at Sinai, and Mohan at University of Michigan for all of their help.
We'll now address some frequently asked questions before going to the chat box. So Anna, what are some of the advantages of using Protrack over other Heatmap tools?
That's a great question, and I think it's a really important one. And so one of the best tools out there for Heatmap visualizations and specifically looking at cancer molecular data is cBioportal. And so I wanted to show sort of the difference in using cBioportal, which is a great tool that I highly recommend, and why Protrack is a more accessible version of this. I'm here on cBioportal, and I want to search a CPTAC study. First of all, it's still quite limited what CPTAC data is available on this platform, whereas we are producing one of these portals for multiple cancer types as we go along in CPTAC. And so if I look at this colorectal adenocarcinoma, it's pretty much the same querying process where you query by gene, and I can select the protein level z-scores, so they do indeed have some proteomics data. You can add in predefined gene lists. If I submit the query, what ends up happening is you also get a really nice interactive heat map that you can sort. To that extent, it's not really that different. And we can add here the protein level z-scores. But the primary difference is that the data right now here is limited. You can't really see the methylome data, the phosphocyte abundance data. The other thing that is the limitation here is if I just took your data and uploaded it to this particular platform. So you could click visualize your data here and then click OncoPrinter. It's actually still very limited what you can actually do. What you end up losing when trying to upload data to really general platform is a lot of granularity. So for example, if you wanted to add in your protein expression, you can only really have the categories of high or low, and you really lose a lot of the numerical differences between them. And so this is an example of why this exists, is so that people can have access to this data in a really granular way.
Okay, thank you. And what other cancer types will this tool be developed for in the future?
This has already been developed for pediatric brain tumor, which is a special study that CPTAC did, which is across 7 different cancer types of ependymoma, medulloblastoma, ATRT, craniopharyngioma, high-grade glioblastoma, ganglioglioma, and low-grade glioblastoma. And then we'll also, in the near future, be developing it for lung squamous cell carcinoma and head and neck cancer. So those are the next. Pediatric brain tumor is already done, and as that paper is published, this will become a public tool, and then the other 2 will come out as the data sets are released or published.
So now we will address some of the questions in the chat box.
Okay, great. So we have this first question from John. Can you apply customized hierarchical clustering dendrograms? No, you cannot, but that is a really great idea, and I'm going to add that as a feature to try to implement. Part of this is that it's also a way for me to get feedback as we're developing this for more cancers. So thank you. That's a really great idea. Is this pulling data directly from a particular data source via an API? Yes, indeed it is. The data itself is actually hosted in a database like the proteomics, the molecular data, but some of the data is pulled down from an API. Specifically, the histological images are pulled out from the Cancer Imaging Archive. The clinical and demographic annotations are from the Proteomics Data Commons, and the gene information is from the NCBI. Another question, what type of cancers? So this particular portal, there's an individual portal for each tumor type. And so this one in particular was clear cell renal cell carcinoma. And then you also asked, can we choose tumor type? Yeah, eventually that's the goal, is as these are produced, then you will have one portal where you can just drop down and select which tumor type to look at. Can the user add additional data points that aren't included in the base data set? You can't, but that's another really good feature idea where you can add, like, say, just a single track with a track name, a series of values, and maybe some color scaling for it. Where can you find the tool you mentioned? I can already download the raw data for the pediatric brain tumor study you referenced. Do you know when the paper is to be published? I do not know when the pediatric brain tumor paper is to be published. I know it has been submitted, so it should be pretty soon. What is the best portal for most up-to-date proteomic data from CPTAC? I would say that the most up-to-date source would be the Proteomics Data Commons, and you can access it by going to cptac-data-portal.georgetown.edu. This is the pediatric brain tumor, and essentially it's all of these tumor types, and you can zoom into individual tumor types to try to understand if you wanted to see them independently of each other or together. And this is the best place to get CPTAC data. It's the CPTAC data portal.georgetown.edu. So Lily asked, how do you open a CPTAC data set in a Heatmap viewer from the data set? Once you hit the viewer, once you go straight to the URL, the CPTAC data set is already loaded and being automatically pulled from a database. So all you have to do, once you have that CPTAC data set, is just pass in the genes that you're interested in, and the data will load up, the heat map will generate, and you'll have a table that you can download. So you just go straight to the URL. There's no uploading necessary. Okay, great. As always, if you have any questions, please do not hesitate to reach out. I really appreciate the feedback with respect to users and also just telling me what features that you would like to use. This very much started off as an internal tool that, as they were writing the paper, they felt was very helpful, and so we're hoping that it can also help other researchers. So if you use it and find ways that it can be more helpful, then by all means, please reach out to me via email at anna.calinawan@mssm.edu.
Okay, Anna, thank you very much for discussing the tool with us and answering all the questions.
Thank you for having me, and thank you all so much for coming.
Thank you for joining the Office of Cancer Clinical Proteomics Research webinar series. Please visit our online resources included here and take a minute to answer our online survey. The Office of Cancer Clinical Proteomic Research is part of the U.S. Department of Health and Human Services at the National Institutes of Health, National Cancer Institute. For more information about OCCPR, please visit www.proteomics.cancer.gov. For general information about cancer or clinical trials, please call 1-800-4-CANCER.